everyone, and welcome to our panel session. We've got some great speakers lined up for you. The panel is Advances in Novel Discovery Tools and Delivery Systems for Aging Science Applications. Just to run through a couple of really quick housekeeping points before we start. Obviously, during the session, please do ask any questions in the chat function. I'll make sure that they are put to the panelists, and hopefully we will get a chance to obviously get them answered for you. I am, however, very aware that no one has come to listen to me today. So without further ado, I will hand you over to our fantastic moderator, Siham, who will introduce the panelists. So over to you.
Thank you so much, Becky, and welcome to everyone. In our session today, as Becky has indicated, we're going to explore together some novel discovery drug discovery tools, as well as some delivery systems that are applied and advancing aging science today. And it is said that innovation is what everyone has seen, but it is also what nobody has thought. And clearly, today in the panel, we have a number of people that thought about things that we haven't been aware of. So we are extremely excited to meet and hear about what they are doing in their respective biotech companies. So I would like to start with a brief introduction. So I'm Siham Ceballos. I'm delighted to be the panel moderator today. I'm the founder and the managing director of Bioartemis. It's a consulting company dedicated to building venture, drug development, and medical affairs. I have spent more than 15 years at different roles in big pharma, biotech, and VC-backed startups. And I hold a PhD in cancer, molecular and cellular biology, and I have spent some time as a scientist on cell senescence in the Ludwig Institute of Cancer Research in London. So it is my pleasure to introduce you to the members of this panel. And first, we have Michael Zemel. Michael is the founder and the chief scientific officer of Nusirt. Michael is a very well-recognized expert in endocrine and nutrition regulation of the metabolism. I believe he focused mainly on fat cell metabolism regulation. And he's developing today, currently, a new kind of drugs that gonna tackle some of the major diseases of our century related to nutrition, namely obesity, diabetes, NASH, and other challenges. And so welcome, Michael.
Thank you.
And we have also Christopher Trummer. Christopher Trummer is the CEO and the CTO and the co-founder of Celeris Therapeutics. He is an expert in artificial intelligence, deep learning, and chemistry or medicinal chemistry. These are all game changers in the drug discovery. So Christopher simply aims to drug in the undruggable, and we would like to learn more about the technologies that he's using in Celeris. Welcome, Christopher.
Thank you very much, Siham.
And we have also Carolina Reis Oliveira. So Carolina is the co-founder and the CEO of OneSkin. It is a female-managed biotech, right? And Carolina holds a PhD in immunology. She has also worked on stem cells, and she switched to Asian biology when she moved from Latin America to Silicon Valley, I believe. And she's now working on some very disruptive rejuvenation technologies. We are very excited to have you, Carolina, and looking forward to hear more.
Thank you
And
so much, Siham.
You're welcome. And we have finally Hank Garcia, who is the head of R&D of Oisin Biotechnologies. So Hank holds a PhD in cancer genetics and molecular and cellular biology that he earned from Roswell Park Cancer Institute. I believe you have also an expertise in neurobiology and science of aging that you have earned as a postdoc in the University of California in San Francisco. And Hank is developing today a DNA targeted senolytic, which sounds fascinating. Welcome, Hank.
Thank you. Happy to be here.
Okay, so let's get started. We're going to focus for the first part on the novel advancing tools for drug discovery in Asian science. And I would suggest that we start by Celeris. So Christopher, you are, I believe you are using today cutting edge technologies like deep learning in drug discovery. And I think that you are aiming for the perfect drug. So could you please tell us a little bit about your company and the tools you are using?
Yeah, it's a pleasure to do so. So Celeris Therapeutics is a deep learning company based in Austria, in the second biggest city of Austria, called Graz. And yeah, we aim to degrade the so-called undruggable targets that you already mentioned, because there's 80% of all pathogenic proteins that cannot be sort of inhibited by all of these modern drugs, such as small molecule inhibitors or biologics. And yeah, most of these targets, they are of course related to diseases everyone knows, such as Alzheimer's and various types of cancer, for instance, prostate cancer and breast cancer. And yeah, there is a certain method known as targeted protein degradation that is actually able to deal with those undruggable targets. Yeah, and in the end, what we strive for is designing novel protags and molecular glues, which are considered to be degraders, and they do that by hijacking the ubiquitin proteasome system. That's what we're doing. Yeah, maybe with regards to what you mentioned about cutting edge and so on. So deep learning and machine learning is, of course, a very fast-pacing environment, right? And many of the methods that have been considered to be cutting edge around 4 or 5 years ago have in the meantime become sort of traditional machine learning approaches. So you really have to yeah, strive for the best research and for novel innovations in order to kind of provide the biggest value to yeah, big pharma and also to humankind in the end. And that's what we're doing. And yeah, the technology that we're focusing on is so-called graph representation learning, because that's the only way in order to design proper protag and molecular clues from our perspective, at least.
Okay, thank you so much. Very interesting. I'm going to turn to Michael. Michael, do you use or did you use this kind of technologies in your drug discovery, or did you have other technologies to share?
Well, thank you. No, our approach was decidedly more manual and more low tech. It would have been delightful to use something, to use a high tech and a high throughput approach, but we started as a university spinoff. Give you a little bit of background. We developed Nusirt with a vision of extending health span, as well as treating the basket of diseases that we would consider diseases of aging and overnutrition. So, or aging and Western lifestyle, if you prefer. I just like the sound of overnutrition better. And the goal was to do this using well-characterized compounds with established safety profiles to optimize energy sensing biology, primarily sirtuins, but also other pathways that converge on the sirtuin pathway, such as AMPK and eNOS. So to do that, we started with our initial discovery, which was that the amino acid leucine is an allosteric activator of Cert1 and of Cert3. And it is unique in serving that role. That is, no other amino acid does so, not other branch chain amino acids, nor non-branch chain amino acids. And so the goal was to find partner compounds. You would consider leucine that as something that would prime the sirtuin system and certainly produce some interesting biology, but not enough biology to be considered a robust drug. So the goal was to find partner compounds. To do so, we wanted to look at existing compounds, starting with compounds that we knew full well would activate the sirtuin pathway and look for synergy there, which we did. And then to build out from there by screening compounds that were well known and we considered to be likely to activate the sirtuin pathway. And again, look for synergy. And we did this fairly simply. We used the seahorse system to assess mitochondrial function. So certainly not high throughput, but not ridiculously low throughput either. So we started with the seahorse system, and over a period of time, and not a terribly long time, I would say about 6 months, over about a 6-month period, we identified classes of compounds that had structural features that enabled us to identify structural features that would predict the next group of compounds. And so once we had our initial 6 months, we were able to drill down fairly quickly by predicting compounds that fit the same structural features, tested them for synergy. And again, we're starting not with new molecular entities or new chemical entities, but we're starting with well-known compounds with well-established safety profiles so that we could move them very quickly through discovery, preclinical, and really fairly directly into clinical. We jumped from non-clinical directly into phase 2 trials.
Wow. So that's thanks probably to the well-known compound, the well-known safety of the compound you were using.
Exactly.
When you talk about synergistic effect, how much do you increase the leucine effect?
So to give you a little bit of background on leucine, this entire story started, and I don't want to take too much time, but this entire story started during my academic career when we were looking at the well-known effects of leucine on mTOR, mTORC1, the sorts of things that we at this conference would very much like not to be activating, and leucine was a well-known activator of. And I just had a vague curiosity, it was really just a side trip of, well, where does the energy come from for leucine to engage, to stimulate all of these anabolic effects? Does leucine stimulate some sort of catabolic biology? And the answer was yes. It was a pretty interesting stimulator of fat oxidation. And so as we drilled down as to where that came from, using a series of rather well-worn and standard approaches, knockdowns, et cetera, we found Cert1, and then using purified Cert1, we showed that leucine was indeed an allosteric activator of Cert1. Now, backing out a little further, how much leucine does it take? So this is where it gets interesting, because if it took the same amount that would be in a meal, well, you would just have a lot of background noise. So let me throw you a few numbers to give you a sense, and then I'll answer your question. Fasting leucine is about, give or take, 0.1 millimolar. If you have a really good high protein meal, you're going to boost up to 0.25, maybe 0.3 molar millimolar if it's a lot.
Right.
We need 0.5 millimolar to fully activate Cert1. Now, how much does it take to activate mTORC1? Well, more than twice that, closer to 3 times that. So in fact, the levels of leucine that we achieve don't touch mTORC1. We've looked at this multiple times, and they simply don't touch it. So now we have this level of leucine that produces interesting biology, but not enough to develop a drug. What we're looking for is a non-additive synergy, a true synergy. So we're looking for something that is going to have fold effects when combined with leucine, 2, 3, 4, 5-fold effects in order to make it a meaningful effect.
Wow, that's amazing. Wonderful. Okay, so let's move to Carolina. Could you please describe your discovery tools and the process and the strategy also?
Sure. So maybe a little bit of background. We started because we have a background in stem cell biology and tissue engineering. We were initially growing human skins in the lab and using those skins to test the anti-aging products in the market, try to validate the efficacy of those products. So at that time, we saw, the beginning of the use of algorithm, using epigenetic markers, molecular clocks that assess our biological age. And then we thought of applying, those markers as a way to measure if, a product could change and could reverse, and we could quantify, the effect of a product by decreasing the biological age of the skin. In that process of testing products that were out there in the market, we basically realized that none of them, they were designed to really target what we believe that was driving the aging process in the skin. So we saw, a lot going on in terms of, tackling the hallmarks of aging, and cellular senescence was gaining a lot of attention. And then we saw an opportunity to really apply that approach to develop a product that was designed to target one of the main processes that's, driving skin aging. So
are you referring to Horvath methylation clock?
Yeah, so initially we used Horvath methylation clock that at that time was only the pen tissue algorithm. So, a combination of all tissues. We realized that we needed a more specific algorithm focused only on skin, because tissue specificity really affects the accuracy of the prediction. So we decided to build our own skin-specific algorithm, to measure more accurately the effects of, those products. And eventually we decided to basically screen for new molecules that would target senescent cells in the skin, mainly focus on the prevention side. So our technology is twofold. One, we are able to measure, to quantify the effect of any compound or any product and how it changed the biological age by assessing the methylation pattern. And the other one is how we screen and how we test our candidates. So because we were growing human skin, we would also be able to replicate the skin aging process. So in the end, when we had the candidate, we would test in aged skins and not, in neonatal skin. That's what is still used for, by the industry today, to see the effect of that candidate in reversing the biological age. And I believe that the fact that we are using 3D models that are more accurately representing, the tissue that we are treating, and also using primary cells, also always trying to get as close as possible to the condition that we want to treat in humans. And using this method that's quantitative to really validate the efficacy of that compound. I think this was a very interesting, combination of techniques that helped us really translate the in vitro data to, when we ran our clinical studies, we could see a very good correlation. So
Wonderful.
basically we end up with, you know, a new peptide that we screen, then we validate, you know, through different assays, and right now this peptide is in our topical product that we launched.
Excellent.
Yeah.
Thank you so much. Maybe Hank, you're in another system anyway. So could you share with us your drug discovery process?
Yeah, sure. So at Oisin, you know, we kind of wanted to tackle aging, you know, from the perspective of kind of imagining, you know, information is basically the language of biology. There's an underlying information, and that's genetics, right? And so previously, you know, a lot of other small molecule approaches weren't kind of getting the specificity or targeting that you know, that could really be able to pinpoint and kill a senescent cell, say right next to a normal cell. And that's kind of what drove the initial idea of Oisin. And so our CEO started as a computer scientist, and so that's why he has this informational genetic approach, and that very much appealed to him, Matt Schultz. And so we partnered with, or he founded the company with John Lewis, who has this exquisite gene delivery technology, which basically, because of the combination of these two approaches, the information approach and the ability to actually deliver that information throughout the body, which had previously been very hard to do, or, you know, inefficient, you know, say AAVs, other lipid nanoparticles. And so by, you know, joining forces, we were able to, you know, first off develop a senolytic, essentially based off of, you know, we owe a lot of what we know now to, you know, we stand on the shoulders of giants, of, you know, like people who have done this amazing work in transgenic mice and other systems, showing that the possibility is there, that we can actually, like, that there's a there to follow. But obviously we're not transgenic mice. We cannot, you know, transduce us up until you know, now, where we kind of discovered that with this gene delivery technology, we can somehow approach the level of distribution of this genetic information. And so that, so we've started with senolytics because we know there's a there. But I think, you know, Oisin is thinks of itself as kind of a gene therapy technology, or a gene therapy company to tackle age-related diseases, and, you know, more just than senescent cells. But obviously, senescence is a great target to start with.
So can you tell us how this gene therapy works? What's the mechanism of action?
Sure, it's very as I alluded to, you know, the people at the Buck Institute and the Mayo Clinic, you know, developed these INK-ATTAC or 3-MR mice to basically able to, upon the P16 promoter, which is a canonical senescent cell gene, essentially drives some kind of toxin depending on the mouse model, and by activation of that toxin, or, you know, expression of that toxin, you're able to only clear senescent cells and leave normal cells untouched. And when they followed these mice out, they saw that, you know, there were improvements in health span, frailty markers, kidney markers, you know, across the board. There seems to be, you know, this effect of senescence that's driving aging, and the removal of those cells helps to not only slow down the progression, but in essence almost rejuvenate the mice, seemingly. And so by being able to take that essential idea, we have our own DNA plasmid, which encodes a P16 promoter driving an ICAS-9, and then upon activation with the dimerizer, we're able to clear senescent cells in effectively the same way, but in humans.
Excellent. So you are still in mice for now.
We're still in mice, but we've also done tolerability studies in non-human primates, canines. I think now the species we've tested are rats, hamsters, you know, in general with our PLV technology. And so, you know, we're hoping to, our first aim that we're trying to go for senescent cells is chronic kidney disease. And so we're you know, putting together preclinical package for that, with our sites on the clinic. And in
Yeah.
general, I'll just comment that the PLV technology itself is already in clinical trials right now, but for a different application, which I won't speak about.
We have a question for you, and it's a great transition for our second part of the discussion, which is the delivery systems that you have worked with. And this question is basically for you, Hank, from the audience saying, can you elaborate on your gene delivery? I assume it is based on modified AAV, and I think it's not. So could you please respond?
Sure. Yeah, so it is not based on modified AAV, which, the downside of AAV-based delivery technology is, generally it's hard to, we all have an immune system that recognizes viruses, right? We've evolved for millions of years to do so. And so the downside of AAV is both the size of the cargo as well as the ability to, administer the therapy to the patient multiple times, because your immune system essentially shuts down that serotype. There are also a number of side effects of your body recognizing a virus, as we're all acutely aware of. And so our gene therapy is based, it's called a proteolipid vehicle. And if you'll let me, I'll just expand a little bit on that. So we call it a proteolipid vehicle, and it's based on a kind of lipid bubble with these fast proteins inside them. And so the fast proteins are very curious, awesome little protein based off of the work of Roy Duncan, a very intelligent and great scientist in Halifax. And so he discovered these orthoreoviruses, which have these tiny little fusion protein inside them. And there are many viral fusion proteins out there, but this one is unique in that it is smaller than the rest by an order of magnitude, and it's very simplistic. All it does is mix lipids together. So by incorporating this protein with our lipid bubble, this proteolipid vehicle is able to effectively promiscuously transduce any cell it comes to contact with.
Great, and only the senescent cells will be killed, basically. That's really exactly. Fascinating. Wonderful.
And just one last thing about it, sorry, is
please.
that because the fusion protein is so small, it is very it's non-immunogenic. We've tried, it's very much non-immunogenic, and because of the heavy lifting of the delivery is done by the protein, not the lipids. We basically use neutral lipids, which on their own would do nothing, but that's good for us because that means that it's safe. So
great, and you have the tox studies so far.
Yes, multiple tox studies.
Okay, shall we move to maybe Michael? Could you tell us a little bit about your drug delivery?
Absolutely. There's actually not a lot to tell. Before I do that, do you mind if I ask Hank a question first?
Yeah, please.
Hank, since you've taken this into non-human primates as well as canines already, is your ultimate goal strictly human pharmacology, or do you have plans to look at lifespan, for example, of companion animals, or is that on your plate?
That is definitely something we can do, and we've had an eye on doing it. I think for us, we're a small biotech, and so you only have so many different directions you can go to at once. Obviously, our main goal is to target humans, but one of the reasons why we test it in canines was because we definitely would. I mean, if there's anyone who would like to partner with us or anything else like that, we definitely have an eye for that because everyone wants to keep Fido alive.
Look, I asked the question because it's a logical. It's a logical extension for anybody who is in the health span, lifespan space to begin to extend the years of our canine or feline companions by a year or two, eminently more testable than trying to obviously extend your lifespan or mine. But you know, I look forward to having enough years to in front of us to test human lifespan as well. To answer your question,
Yes, Michael.
we are very simple. Our delivery system, well, yes, well, it's oral, but again, this is driven by who we are. The fact that we are relying on established patterns of safety, that we are trying to repurpose existing molecules that are well known, well established. In most cases, they are drugs that have been used for other purposes, sometimes at different doses. We tend to go lower rather than higher in our repurposing because we can. In some cases, they are well-known natural compounds for which there is still ample safety data involved. If we start layering a novel delivery system on top of that, we're going to have to revisit safety again. As and we are not eager to revisit safety, not only from a practical perspective, in terms of not wanting to go back, not wanting to go backwards, but simply to enable speed to clinic. We've been very fortunate with each of our INDs that we've been able to go from animal work directly to phase 2 using the FDA's 505(b)(2) pathway, as opposed to spending a lot of time in human phase 1 studies. Obviously, we've had to look non-clinically at interactions between the compounds that we're putting together to ensure that there were no unexpected interactions. But those can be done in a matter of months as opposed to years.
Very smart repurposing drugs from the safety perspective is really great move.
Sure. I'll throw an example at you. One of the drugs that we have repurposed is sildenafil. Sildenafil, of course, is well known as Viagra, but the starting dose of Viagra is 25 milligrams. The goal with Viagra is to be a phosphodiesterase 5, PDE5 inhibitor. I have zero interest in sildenafil's PDE5 inhibitory activity, but at very low doses, not 25 milligrams, but 1 milligram, at very low doses, it is an eNOS activator that converges back on the Cert2-in-1 system because there's robust crosstalk between eNOS and Cert1. So I'll just use that as an example, given the amount of safety data that is available and RX labels up to 100 milligrams of sildenafil. It really is not a heavy lift to persuade that 1 milligram is likely to be safe.
Great. Thank you, Michael. And Carolina, your strategy is very interesting, going as a supplement, a topical supplement. Okay, can you tell us more?
Sure. So I think as everyone here, we are all interested in extending health span. And also because our experience in skin biology, we really thought that skin would be one of the first tissues to benefit from, those therapies that are, designed to target the root cause of aging. And also because of the regulatory simplicity. And once you validate the safety, you can bring, a novel compound, in our case, a peptide it's a biological molecule. It tends to be safer. Of course, we ran all the safety studies, but the idea is that we could already, bring those technologies to the humans and start to get more data and more insights how this affects our tissues. Can we really reverse, our tissues biological age by applying, a topical product And that's basically the strategy that we decided to take. And right now we are running a clinical study that we are gonna measure this, change in the biological age by collecting biopsies and really validating for the first time that, we can reverse in humans, skin biological age, that I think would be super interesting. And in the future, this candidate that we are using for skin can be also explored and applied to other, age-related diseases. And we are already testing these in other models. But I think, skin has its advantage, and I think is a great way to start to, bringing those technologies to consumer early on, once the safety is cleared.
So Carolina, when you measure the clock in the skin, and do you see it slowing down? Do you see it reversed? What do you observe exactly?
Yes, great question. So we've tested in 2 different models in the 3D skin equivalents that we build, and also in skin biopsies. We could see reverse up to 10 years. There is a larger error or variation for the 3D skin models, but we also saw a reduction of 3 years from the control to the treated skin.
After
skin
duration, what duration of treatment?
5 days, but yeah. Yeah, but 5 days was mainly applying the, adding the molecule in the media. So probably the absorption, it's higher than topically, was more like a validation. That's why now the first time point that we are measuring these clinical studies after 6 months. So we believe that, a topical delivery is gonna take a little bit more time. And then we're gonna continue up to a year to be able to detect that change in the biological age.
Do you interpret this reverse effect of aging as a stimulation of stem cells and renewal of the tissue, or just the death of senescent cells?
I think, well, we have both, right? So, once you prevent or you cleared out senescent cells, basically you change the environment that the cells are in. So you decrease inflammation, and this starts to allowing for other cells to healthy cells to replicate and to replace the cells that are dying.
Yeah.
And so by decreasing the level of inflammation, we also see that we have more collagen production. So it affects like different layers, from cell proliferation, from molecules that are secreted. And in the end, this is also reflected on the epigenetic pattern, So it's a very interesting, because we are not really changing directly the epigenetic profile, but the end result is that by addressing one of those mechanisms, we can see a change in the epigenetic profile.
Wonderful. I guess maybe some of the panel members have questions around this. No? Okay.
I know that skin is not a typical area of exploration.
Yeah, but it's extremely attractive and interesting. Aging is the first signs of aging are on the skin, and it's the most accessible organ that we can work on. It's fascinating.
I love the approach of starting with the skin as sort of the tip of the spear, as a safe approach that you can then expand from. I think it's very smart.
Exactly.
Yeah, and the skin is, the biggest organ as well as the most easily accessible. Yeah. So,
And is your product on the market already?
Yes, we launched officially in February of this year. So we're getting very good positive feedback so far. That's very encouraging.
Wonderful. Are you in Europe already? We are looking forward to it.
No, unfortunately, we are working on international shipping, but it's almost there. It's a little, it's kind of, yeah, on the process.
Excellent. Christopher, do you have ideas or comments on the delivery? I know you are more in the drug discovery than in the delivery, but maybe you have been involved in some systems, or you have comments on the rest of the panel's technologies.
No, exactly what you mentioned. So we're really in the early stage drug discovery, so to speak, and not too much in the drug delivery. Anyways, I really liked many of the comments from you guys. What kind of caught my interest was always talking about like prolonging a healthy lifespan, so to say, on the one hand. But there's, of course, that's just I guess one part of the truth, right? So you can try to prolong this kind of lifespan via different types of methods, but still you will always face maybe some sort of diseases within a given life you might not be able to kind of overcome very easily, right? So that's what we are dealing with essentially, like all of these kind of diseases that are really hard to tackle. That's it from my side, of course.
Excellent. Thank you. I'm going to turn to some of the audience questions. So for Carolina, you have another question about have you tested it in the presence of crosslinks, in particular glycospan?
Yeah, that's a good question. Since we use human biopsies, and a lot of times they come from, you know, elderly patients, so they already, you know, they already bring out all of those crosslinks. So we have tested in those conditions. We have skins coming from donors from, you know, 76, 80, 90 years old. So we do believe that there is a lot of, we didn't measure specifically, you know, how much we are changing the crosslink. But yeah, definitely older patients, they bring that condition.
Excellent. Another question, believe for Hank. How did you prove PLV target efficiently the senescent cells?
Well, by essentially, you know, we first validated our process, you know, in a dish, and we were able to do a very cool time-lapse microscopy, wherein we had, you know, effectively, you can think of this as just transfection for those who are laboratory minded. So you're able to, you know, express our therapy with the GFP tag, track the cells, time-lapse microscopy, the active cells or the cells expressing the construct die, the normal cells virtually untouched. We've also validated this in vivo, looking at SA beta gal and P16 expression, and those decrease, you know, after the treatment of our therapy. And we've also tested this specifically in kidney, because, you know, I just want to tack on to what Chris said. You know, it's one thing to increase health span, but, you know, like the I think a goal of all of us is to, you know, to try and reduce unnecessary human suffering. And I think that, you know, these age-related diseases, you know, whether we prevent them, which is very important, or whether we tackle them as they arrive, these are all, you know, great ways to do things. And you know, it's great that, you know, we're trying to tackle age-related diseases, but, you know, Chris is tackling the future of age-related diseases. You know, it's like, I'm sure senescence and senolytics won't be the only approach to tackling the aging process.
What's your first indication, Hank?
Chronic kidney disease.
Okay.
I think Hank just, if I could, I think Hank just outlined the problem that we all face, which, regardless of technology, which is, you know, at the end of the day, you need an indication. We can talk about lifespan and we can talk about health span, and we all have those goals. Fortunately, when we talk about health span, we are talking about indications because we are talking about compressing the disease span. So we are dealing to some extent with treatment of disease. But because every one of us, at least on the RX side, has to chase an indication and then hope that we can find a way towards broader lifespan, health span, preventive medicine for which there are no indications. On the other hand, those of us who also have, as Carolina does, as Nusirt also does, have a non-RX side, we have a little more latitude on the non-RX side in chasing health span. Lifespan is still going to be a problem because, you know, clinical data being what it is and taking how long it takes to collect.
Absolutely. And on this note from Michael, I need to wrap up. So thank you so much, all the panel member. It has been a pleasure and a real nice discussion. I hope everyone has appreciated as I did. Thanks to the audience for joining us, and thanks to LSX for organizing the event. I hope to see you soon, all of you, and maybe some of the attendants during the conference in a one-to-one meeting. Have a nice day.
Thank you so much, everyone. Thank you. It's been great, and I hope everyone enjoyed the panel as much as I did. Sorry to cut the conversation short, but hopefully we'll see you all again soon. Thanks so much.
Thank you, Becky. Bye.
Thank you.
Thank you.
Well.
Thanks, everyone. So we're not live anymore.
Oh, good.
Okay.
Well, it was a pleasure to participate.
No, good.
I hope it went well for you as well.
Yeah, 45 minutes always sounds like a lot, but it goes really quickly. So
I had the impression that we had a little bit more time. So my clock was not indicating the same time that you were giving me. I had more time theoretically, but I tried to stick to your time.
It is 45 minutes. I mean, it always goes really, really quickly when you're doing them. So, but no, it was really great to have you all involved, and hopefully you'll get people sort of, reaching out to you over the platform and things like that. And, because we're all about obviously growing the community. That's what we're here for. So, but I'm sure Angela will be in touch, obviously, because, she's the one that's been producing it and sort of put everything together so fantastically. But thank you so much, everyone. And if you've got any questions, obviously send them over. But very much hope to see you again soon.